Thank you for joining CADASIL Association's fourth webinar in our CADASIL Connection speaker series. I am Jane Gunther and I will be your moderator. Today's feature presentation is entitled Harnessing the Power of the Immune System to Develop Therapies for CADASIL, presented by Dr. Joseph Arboleda Velasquez, known to many as Dr. Joe. I will start today's webinar with a brief overview of the potential biomarkers and therapeutics currently being researched for CADASIL. Before we begin, I'd like to go over a few housekeeping items. We've taken a screenshot of an example of the attendee interface. You should see something that looks like this on your own computer desktop in the upper right corner. You are prompted to select your audio, and the default setting is computer audio. If you prefer to join over the phone, which can provide better audio quality, select telephone in the audio pane, and the dial-in information will be displayed. You have all joined this webinar in listen-only mode. All callers are automatically muted. At the beginning of the presentation, Dr. Joe will answer questions. At the end of the presentation, Dr. Joe will answer questions that were pre-submitted during the webinar registration process. However, at any time during the webinar, we invite you to type questions in the question pane of the control panel. These questions will be forwarded to Dr. Joe following the webinar. Even though these questions will not be answered today, it is of value to know the types of questions the CADASIL community has about their disease. Today's presentation is being recorded and will be available on the CADASIL Association website. In addition, we have a handout to share with links to Dr. Joe's published papers on his personal website. In your control panel, you will see a handouts pane and the document listed that you can download. The devastation that CADASIL had on their families led a group of volunteers in 2012 to establish the CADASIL Association, whose mission encompasses patient advocacy, education, and research support. The dedication and passion of these volunteers to cure CADASIL has led to a number of successes by our association. One of these successes has been our support of Dr. Joe's research that has yielded 2 publications, the results of which are being presented today. And information to apply for a research grant is available by contacting cureCADASIL President Nancy Maurer. CADASIL is a rare genetic disease caused by mutations of the Notch3 gene. Dysfunction of the Notch3 protein leads to its accumulation in smooth muscle cells of the brain vasculature, with loss of the important function of these blood vessels to supply adequate oxygen and nutrients to brain cells. While likely more complex, simply stated, the resulting areas of low blood supply in the brain causes damage to neurons and loss of the important myelin sheath around neuronal axons that is crucial for normal conduction of signals between neurons, resulting in the physical disabilities and cognitive impairment seen in CADASIL. We created the CADASIL Connection speaker series for researchers and clinicians to present their work in CADASIL. It's really a unique opportunity for the entire CADASIL community to learn about the current state of research in CADASIL, in particular new understandings of disease progression and mechanisms, and today's feature presentation, which will focus on the identification of potential biomarkers and a potential therapeutic. Our hope is this series will accelerate research and create collaborations that will make meaningful differences in the lives of those with CADASIL. Already, the series has been a useful tool as we direct interested companies to the recordings in order for them to learn more about this disease. I will start today with a short introduction to potential biomarkers and therapeutics for CADASIL, and I'll be followed by Dr. Joe's presentation. I wanted to give you a little introduction to myself and why I'm interested in biomarkers and therapeutics in CADASIL. I worked 8 years developing biomarkers for the clinical laboratory. Wanting to do more research-related work, I returned to school and completed a PhD in neuroscience from the University of Utah, where I researched the mechanisms of epilepsy. I continued researching signaling mechanisms of neurons as a postdoc at the University of Washington. I returned to the pharmaceutical industry, where I directed efficacy and toxicity studies for novel drugs and often challenging diseases. I'm a board-certified toxicologist and have worked 17 years as a non-clinical toxicologist, creating the safety strategy and performing the tests necessary for new drugs to receive FDA approval to enter into clinical trials. I've worked on drugs for multiple sclerosis, Alzheimer's disease, and numerous immune-mediated drugs or diseases. Currently, I'm a director in non-clinical toxicology at Nectar Therapeutics in San Francisco. I have worked on many drug programs and seen very few make it to a market approval, either due to failure to show efficacy in the clinic, even though efficacy was seen in animal models, or due to a safety issue that did not occur in the animal studies. In recent years, more effort is being made to identify appropriate disease and safety biomarkers in order to increase the chance of success of our drugs. I've been an active member of CADASIL Association since 2016 and a volunteer trustee since 2017. My boyfriend and his family have CADASIL. I'm grateful for all the work CADASIL does to championing the families and the development of therapeutics for CADASIL. And one thing I've learned by volunteering is in the rare disease world, patient organizations play an important role in drug development for their disease. If you have questions or ideas on biomarkers or on therapeutics, please email me. My email is here on this slide. A remarkable amount of progress has been made since the last time this slide was presented at our first webinar in April of 2017. While there are no treatments and no clinical trials at this time, there has been great progress made in development of potential biomarkers in Dr. Joe's lab, which she'll be presenting today, and by the MARC VCID Consortium, which recently held a meeting to kick off year 2 of their 5-year goal to identify and validate clinical trial-ready imaging and fluid biomarkers for diseases like CADASIL. What are the potential CADASIL biomarkers? There are 2 that are validated and used to diagnose that a person has CADASIL. One is a genetic analysis that identifies a NOTCH3 gene mutation associated with CADASIL, and the other is identification of GAM, a deposit on small arterioles that can be seen using electron microscopy of a skin biopsy. Currently, in order to follow CADASIL disease progression or help determine efficacy of a therapeutic, there are only potential biomarkers in the research and development stage. Dr. Joe has published on 3 potential target engagement biomarkers and will be presenting this encouraging data for these 3 proteins that are found in the blood. In addition to fluid biomarkers, disease progression may be monitored using a functional readout such as MRI to measure brain blood flow, or by monitoring brain changes using imaging techniques. A combination of biomarkers may be necessary to monitor disease progression and to assess the effectiveness of potential therapeutics. What is desired are biomarkers with specificity, that is, biomarkers that are specific to the disease and can identify it as one disease from a similar disease. And biomarkers with sensitivity, biomarkers that can measure small changes in the disease. And one of the benefits of this is it could make it possible to shorten the duration of a clinical trial. Biomarkers with high specificity and sensitivity will help in the identification of therapeutics that have higher probability of translating into successful treatments for CADASIL. CADASIL is aware of 4 potential therapeutics in the research proof of concept stage. At our August webinar, Dr. Zhao presented data on bone marrow stem cell factors, stem cell factor, and GCSF. They were found to increase cerebral blood vessel density and improve cognitive function in a mouse model of CADASIL. Her student will be presenting new mechanistic data with these stem cell factors in the CADASIL mouse model at the American Society for Neurotherapy and Repair Conference in April. CADASIL is sponsoring a travel grant for a student to travel to this conference, and this has been supported by donations from our board. The second potential therapeutic is an oligonucleotide that binds to the defective region of the NOTCH3 gene, and it allows a read-through of the gene without including the mutation, such that a normal NOTCH3 protein theoretically is generated. Dr. Saskia Oberstein's lab demonstrated that they successfully had Notch3 correction in a study performed in a culture dish with cerebral vascular smooth muscle cells obtained from a patient. Dr. Anne Joutel's lab has reported positive results with an antibody that binds the mutated regions of the Notch3 protein and prevents disease in a CADASIL mouse model. The fourth potential therapeutic is today's topic. Dr. Joe will discuss research performed in his lab using antibodies to target mechanisms involved in CADASIL pathology, including defective Notch3 signaling and Notch3 protein accumulation. The biomarker and antibody research that will be presented today was supported in part by donations from cureCADASIL Association. I'm pleased to introduce Dr. Joseph Arboleda Velasquez. Dr. Joe was introduced to the field of small vessel diseases as a medical student in Colombia in 1997, where he worked under the supervision of Dr. Francisco Lopera in the clinical characterization of two large Colombian families with CADASIL. With his medical degree completed and a goal to become a research scientist, Dr. Joe joined Dr. Kenneth Kosick's lab at Brigham and Women's Hospital as a research fellow. In Dr. Kosick's lab, he researched the genetic characterization of the Colombian CADASIL families and conducted experiments on the molecular characterization of CADASIL mutant receptors. Dr. Joe completed his PhD in the lab of Dr. Spiros Taconis at Harvard Medical School, researching the role of Notch3 signaling in vascular smooth muscle cells and susceptibility to ischemic stroke in animal models. He generated and characterized animal models of CADASIL that closely reproduced the pathology observed in humans and allowed for the characterization of the vascular deposit that is found in CADASIL patients. To broaden the scope of his research, Dr. Joe joined the lab of Dr. Patricia Demore at the Shepton's Eye Research Institute as a research fellow. Dr. Demore has been at the forefront of research in mechanisms of vascular growth and development. Dr. Joe studied the role of cell-cell interactions in diabetic retinopathy. He was appointed faculty investigator at Shepton's and currently is assistant professor at Shepton's and Harvard Medical School. Dr. Joe's research encompasses two areas: pathobiology of small vessel diseases affecting the brain, specifically CADASIL, and the pathogenesis of diabetic retinal disease. Dr. Joe recently co-founded the drug company Vastora and is on the scientific advisory board of cureCADASIL. Welcome, Dr. Joe. We look forward to hearing your talk on harnessing the power of the immune system to develop therapies for CADASIL.
Hello, everyone. Thank you so much, Jane, for your very kind introduction. And I also want to thank everyone that is attending this seminar and cureCADASIL for organizing this very interesting seminar series. The title of my talk is Harnessing the Power of the Immune System to Develop Therapies for CADASIL. I first want to disclose, as Jane mentioned earlier, I am a director and co-founder of a pharma and biotech company, Vastora. This is in addition to my academic endeavors. And also I'm listed as an inventor in patents filings for blood biomarkers of a small vessel disease and also for Notch3 signaling manipulation as a therapeutic modality in a small vessel disease. This work that I'm gonna share with you today, I would say was really inspired during almost 20 years ago when I was a medical student back in Colombia. In my home city of Medellin and in the vicinities of the city, there are many large families with CADASIL, and When I was a medical student, I had the chance of seeing this patient that made a profound impression on me. She had 47 years of age, and she suffered from a stroke and came to the emergency room. Back then, I had decided already to focus on research and was learning about clinical research at the time. She had history of migraine with aura, um, and she had suffered previously from a stroke at age 24. Um, Intriguingly, like in many other CADASIL patients, she had no cardiovascular risk factors, no diabetes, no hypertension, but she had suffered already from like at least 2 strokes. Um, um, and what was impactful to me was really that uh, in medical school we learn about this strong association between diabetes, high blood pressure, and cerebrovascular disease. But in this patient, that was absent, and it was also absent of several of her relatives that also suffered from stroke. Uh, in working with this woman and her family, I got to really spend a lot of time trying to, recruit many of the family members for our research study. It turns out that we were able to put in the family genealogy for this particular family almost 300 individuals. The family was very large, and then that was in addition to another CADASIL family where we had put together a pedigree of over 1,000 individuals. Many people here in the United States, they really have, just small nuclear pedigrees, but in Colombia, the demographics are such that you can actually identify these very large pedigrees, and we're able to track the disease in many of these patients. What I'm showing here is an MRI of this individual that, as you know, has these characteristic abnormalities in the subcortical region, and this is the way it should actually look in a normal individual. This is a story that for me began with this patient in Colombia is really a global problem. CADASIL is actually the most common cause of inherited cerebral small vessel disease. And we have always thought of CADASIL as very rare, and we also thought the mutations were very rare, but it became very clear that the mutations could be very frequent. NOTCH3 mutations that had been shown to cause CADASIL turned out to be about 100 times higher than we previously anticipated. And the prevalence in the general population could be as high as 3.4, about 3 individuals per 1,000 in general population. Now, that doesn't mean that everybody that has the mutation will have the disease, but certainly they are at risk. As I mentioned earlier, the CADASIL phenotype is really characterized by this adult onset stroke. As a consequence of the strokes or the brain ischemia, sometimes the strokes are not symptomatic, but people start losing cognitive abilities. So there is this could become progressive and lead to dementia. And as I mentioned earlier, the absence of cardiovascular risk factors. The disease is autosomal dominant. Usually you have one affected parent, and half of the children are at risk, at least half of the children are at risk of getting the disease. As I mentioned earlier, we were able to identify many relatives from this Colombian family. It has been shown that CADASIL is linked to mutations in NOTCH3, and this is why we call CADASIL a monogenic condition. All the mutations that have been described as caused in CADASIL are in this gene called NOTCH3. Now, I wanna focus on a particular feature of of CADASIL pathology that I think is important for us to fully understand, more so in degenerative conditions like Alzheimer's, even in Parkinson's disease. There is a very strong focus on some pathological abnormalities that are called deposits. And the deposits are very important for neurodegenerative disorders because they could be diagnostic. For instance, CADASIL has its own type of deposit. It's called the granular osmophilic material. If you look at the vessels of a CADASIL individual, this is a vessel is made of a lumen, endothelial cells, and then you have a layer of cells that are the small muscle cells or the pericytes that provide support to the vessel. Well, in CADASIL, instead of having these small muscle cells and pericytes, what you have are these deposits that, if you look at them by electron microscopy, they look black. So they gather a dye that is used in electron microscopy called osmium. So that's why it is called osmiophilic. And these deposits are very important, as Jane was mentioning earlier. They could be even diagnostic for the disease. But I want you to pay attention to the fact that, well, the deposits are there in the place where the small muscle cells and the pericytes should be. So, and this is what I'm showing you here. In this slide, you see the way a vessel should look in a normal brain. And this is the staining for the small muscle cells that provide support to the vessel so that it doesn't collapse and the blood can flow through it. But in CADASIL, those cells are gone. And this process is progressive, and it leads to a weakening of the arteries. And when the arteries are leaky, they can collapse, they don't function, they don't provide the blood flow, and they can also have hemorrhages. And I want to focus on this because sometimes I think there hasn't been enough attention paid in the CADASIL field, perhaps because of the lack of models in the loss of the small muscle cells. And this is very important because there is a key characteristic of CADASIL, which is a strong correlation between the expression of the gene that is mutated in the tissue and the pathology. NOTCH3, the gene that is mutated in CADASIL, produces a protein that is expressed highly in the small muscle cells, and those are the cells that die, which give you, other cells like the endothelial cells, they don't have as much, or the neurons, they don't really have as much NOTCH3. Small muscle cells are the ones that express it and the ones that suffer. Now, what is notch? Notch is a very important signaling mechanism, and that cells use to communicate with each other. The notch receptor is expressed in one cell, and the ligand that is recognized by the receptor is expressed in another cell. So the moment that the two cells are in contact with each other, notch interaction with the ligand can occur, and that triggers a series of cleavages of the notch receptor, leading to the translocation of the intracellular domain of notch to the nucleus, and that regulates transcription. We call this gene expression. And this mechanism of signaling is quite conserved across species, and is used once and over again in different developmental processes for the cells to communicate different messages. For instance, cells use this to communicate the message of coordination for proliferation, or coordination for cell death, or coordination for growth. Now, it is important for me to mention that although there is a strong correlation between NOTCH3 signaling and stroke and the small muscle cells, the Notch pathway, meaning there are other Notch receptors and other ligands, have been involved in many different cardiovascular diseases. I'm listing here just a few as an example. Here is CADASIL linked to NOTCH3, but there are other diseases like arteriovenous malformations, pulmonary hypertension, aortic valve disease, and allergy syndrome. Is a cardiovascular condition, all of which have been associated with mutations in other genes that are related to the Notch pathway. And this really, if you look at it, from above, I want you to get the idea that the cardiovascular system vessels seem to be exquisitely sensitive to the dosage of Notch signaling. What these mutations do really is that they either diminish Notch signaling or they augment Notch signaling. And the moment that happens, you have pathological vessels, abnormal vessels. Now, for all these conditions, that has been very clear. For CADASIL, it has been trickier it has been more difficult to determine what is the impact of the mutations on signaling. But I want you to give you the general idea that the Notch signaling levels are very important for vessels. Now, why are the mutations in CADASIL, many of them, somewhat different than other mutations found in other genes leading to cardiovascular disease? But the mutations affect a region of the receptor in the extracellular domain that is called the epidermal growth factor-like repeats. And in each of these repeats, you have an amino acids that are called cysteines that have this ability to form these disulfide bridges that fold the repeat like an accordion. Well, as it turns out, in CADASIL, many of the mutations, really most of the mutations, lead to the loss or the addition of one of these cysteine residues, which means that now you don't have these nicely paired cysteines, and instead you're going to have an odd number of cysteines, either 5 or 7. And these cysteine residues are very reactive. They like to really stay in the bridge configuration. So it has been proposed that, and there is some evidence for that, that these reactive cysteines can trigger the formation of dimers between proteins that shouldn't be there, or between Notch 3 perhaps with itself. I have published some evidence of that, or between Notch 3 and other proteins. We have published, for instance, a heterodimer with French. Now, there are many CADASIL mutations reported, and most of them impact cysteine residues. But the gene is made, and the protein is made. It's just that it has that one defect, and that perhaps capability of forming aberrant interactions. So it has been shown that some of the receptors retain signaling capability, and some of the Notch receptors carrying CADASIL mutations do not have signaling capabilities. And we don't really know exactly what's the relative contribution of each of these mechanisms to the disease. Could it be that this protein-protein interaction is more important than signaling, or the other way around, or the two of them are happening? And this is a field of active research investigation in the field. So this is what we call molecular genetics, the mechanism of the disease, how the mutations lead to the phenotype, and this is still unresolved. Now, why is molecular genetics important? Well, molecular genetics is important because the moment you know the mechanism for a disease, you can design rational therapeutics. Well, if the disease is because the gene is not working very well, maybe we should have a drug that activates the receptor. Or if the receptor is hyperactive, maybe we should have a drug that tames the activity down. Or if it's really like a problem of protein-protein accumulation, well, maybe we should have a drug that prevents the interactions from happening, or that leads to clearance of these deposits. Now, the first step is really to have a good idea of a good hypothesis, and I think in the CADASIL field, we have already made some progress that show us what's the potential mechanism for the disease, and all of these are hypotheses that have some evidence, some supportive evidence. Now, the moment that you have these hypotheses, then you need to test them, and to see if by counteracting the mechanism, your proposed mechanism, well, you can have a beneficial effect. Now, how do you know a drug works? That's where you have biomarkers, and Jane explained very well how these biomarkers work. They essentially tell us if a drug has the potential to be working. And when you have it, you can actually make a very efficient clinical trial. And then obviously what you need a therapy to test. And all of this is something that is an active area of investigation and that we have like focus on in my lab. Now, what is the insight for my research, as I told you, had to do with that Colombian patient. What I found intriguing about her case was that in this individual and her family, we found this mutation, the same sorry, I think there was a blackout, but I think I'm back. For the, let's see if this is correct. I think I'm fine. For the the insight that came from this individual was that in her family, the disease started quite early. Usually they got their first stroke when many of these patients were in their 30s. And that does not mean CADASIL causes early onset of a stroke, but really in most families what you find is that the strokes in average start when the patients are in their 40s and 50s. So right there we were able to establish like genotype-phenotype correlation where patients with this mutation had an earlier onset of a stroke, and the patients with this old mutation, and in general, most CADASIL mutations, a later onset of a stroke. And the insight came from this observation that the mutation that had the early onset of a stroke, that is happened earlier in life, were located in the ligand binding domain. Specifically, although the receptor is very large, these two epidemic growth repeats are the ones that are necessary and sufficient for the receptor to bind with the ligand. So quite obvious, we thought if the mutation that is in the ligand domain it perhaps impacts signaling, leads to early onset, that means that signaling plays a role in CADASIL. We published that in the journal Neurology in 2002, and some aspects of this correlation were extended and in European families and confirmed. For instance, they indeed confirmed that patients that have mutation ligand binding domain had widespread MRI abnormality. This is important when you have a genotype-phenotype correlation that we demonstrated in a Colombian family that's corroborated, at least in some aspects, in individuals with different ethnicity. Now, again, if we have this idea that there's a role, so then, and the idea is when you have less signaling, you have a phenotype, then one can propose, as you mentioned earlier, this modality of treatment, in which we can say let's actually restore normal NOTCH3 signaling in these CADASIL patients, and let's see if that could help with their disease as a treatment. And what's exciting was that NOTCH, because NOTCH is so important for conditions in development, in cancer, there were a lot of companies that have been interested in developing drugs for NOTCH or that we target NOTCH receptors. And indeed, we knew that some antibodies that specifically recognize had been developed and had the ability to either activate NOTCH3, in this case the A13 antibody, and or inhibit NOTCH3 signaling like the A4 antibody. We have some tools available to modulate NOTCH3, and this is where this idea of harnessing the power of the immune system comes about. Well, antibodies are essentially a tool from the immune system that helps us fight disease, and we make antibodies for against bacteria or against viruses to help us fight disease. So something that makes immune system so useful for therapy is that the immune system is very specific. if you wonder why you have to get the flu shot every year, well, that's because every year there is a new strain of the virus that is the flu that is a tiny little bit different than the one from last year. So you need to be immunized, get the flu shot to develop antibodies that would be specific for that strain. So the specificity of the immune system, and in this case the antibody, is such that you can generate an antibody that recognizes NOTCH3 specifically instead of NOTCH1, NOTCH2, or NOTCH4, we call it NOTCH receptors. And you can make it so that it recognizes specifically a part of NOTCH3, and you can make it so that it has properties in how it interacts with the receptor. And what I want to bring to your attention here is the power of the immune system. A potential tool for us to discover therapy for CADASIL. Here, we're talking about a mechanism in case antibody that has the ability of activating NOTCH3, but there are other mechanisms at play in CADASIL NOTCH, like protein accumulation, and you can think of antibodies that have that property, for instance. So there is interesting opportunity for CADASIL therapy. One is if antibody that is specific, you will bind only NOTCH3, and then NOTCH3 is specifically present in the vessels, and that's very important, quite an advantage. There are other receptors like NOTCH1 that have a very wide expression. So even if you have an antibody that recognizes NOTCH1, well, that's great, but NOTCH1 is expressed in many places. So you can have more side effects. Instead, with NOTCH3, not only you can have an antibody that's specific for the protein, but also then the protein is expressed in the, so you have double. Something important about this antibody, I told you how it could be specific for a region of case. The antibody that we're talking about, the A13 antibody, binds specifically to this region that is inhibitory activity. And when it does, it really activates the receptor. I want to bring to your attention the fact that all the mutations that have been reported in CADASIL are located in the EGF repeats, basically in another region that is not bound by the antibody. And that's ideal because that means that potentially this antibody will have activity on all the reported mutations. If it was, for instance, binding here, it could be that the mutation can affect the receptors such that the antibody does not bind. That's not the case. It binds a different region. And I want you to also appreciate the fact that, you know, it seems like NOTCH here is like broken. It's not really continuous protein. Indeed, in the plasma membrane, the NOTCH receptor is broken. It's what we call a heterodimer. And the dimer is really bound by very weak interactions that are supported by these negative regulatory regions. What the antibody does is that it gets in this area and it dislodges the heterodimer, making this region to be released, and then some proteins that are important for the activation of the receptor will then be able to just activate the receptor constitutively. In this case, there are some proteins like the gamma secretase complex that do that. Now, this drug, this molecule has been, you know, this antibody has been was generated a long time ago, and we honestly had knowledge of the antibody before we could even test it because, as I mentioned earlier, you need biomarkers. Let's say we apply this drug to an animal model or an in vivo model of CADASIL, how do we know it works? That's where we need biomarkers. So although we had the antibody in a way available, we couldn't really do the necessary studies because we didn't have the biomarkers. So in my lab, we had to take a step back. And this is the first project that was supported by cureCADASIL Association. They actually provided the seed funded for the discovery of these antibodies, of these biomarkers, I mean. So to discover these biomarkers, we took an approach. I don't want to focus too much on how the discovery worked, but essentially we use analysis of proteins. We did what we call a proteomic study of CADASIL vessels. In that case, we took vessels that had been donated by, um, from brain tissue that had been donated by Colombian patients. They, when the families, when one of the relatives die, there is the option of donating the brain. I know some of you have been involved in that and the process, and that's very important for discovery. So we had some of this tissue. What we did was that we collected the brains, and we did using this laser capture microdissection from the brain tissue. We can collect the vessels and do an analysis of proteins using a method called mass spectrometry. That was one way in which we made the discovery phase of the biomarkers. The other one was that we had generated some animal models of CADASIL, and we took the blood from these animal models, and we subjected the blood to these arrays that allows us to detect different proteins that could be increasing or decreasing. And then each of the dots here represents a protein, and the intensity shows you how much of that we had. And this led us to identify some of our candidate proteins. So again, this was a combined effort in which we were learning from the human tissue, and we were learning from the animal models, in that case using blood samples. So we're able to come up with four candidate biomarkers, of which I'm focusing here on three, or depending on the case. One of them is endostatin. We found that endostatin was changing, and the levels were abnormal in animal models that had the CADASIL mutation, but not in the control. Endostatin actually is a very important biomarker because we are finding it everywhere, really. We found it that it changes in the blood of the mice. We also found it that it was changing in the brain vessels from the post-mortem human tissue. And then we found that it was actually regulated, the expression of endostatin was regulated by NOTCH3 signaling, and this is what we're showing here. Endostatin is also very important because it's a fragment of a larger protein called collagen 18 alpha 1, and this fragment has very strong anti-angiogenic properties. So we've always been very interested in endostatin because we think it could be a biomarker, but also because we think it could play a role in the pathology of the disease. It's quite intriguing that an anti-angiogenic protein, a protein that essentially diminishes the growth of vessels, is changing in a disease like CADASIL that is characterized by vessel degeneration. The other protein that we found was changing was HTRA1, and again, it was different the levels in the blood of the mice that had the CADASIL mutation compared to control, and we found that in the blood, but we also found that in the tissue. So this protein seemed to accumulate in the tissue, in the vessels that is present NOTCH3 receptor that has a CADASIL mutation. And the interesting thing about HTRA1 is that it happens to be made by the gene that is mutated in another disease. It has a name similar to CADASIL, but it's called CADASIL because instead of being dominant, it's recessive. So it's called CADASIL. It's a recessive form, and that means that both parents have to be carriers of the disease for you to be able to get it, as opposed like in CADASIL where only one parent has to have the mutant gene. So it was quite intriguing that we found here was the first report of a link between these two diseases, like a functional link. It seems like when you have a NOTCH3 mutation, you misregulate the protein that is mutated in another completely different disease that happens to have a similar phenotype. These patients also get stroke and vascular dementia. The other biomarker that we came up with was that one we did not discover via any of the proteomic studies. It was really just kind of thinking, what could be changing in CADASIL? So we reasoned that, maybe we were able to detect the NOTCH3 extracellular domain in the blood. And we knew that the NOTCH3 accumulates in the vessels of CADASIL patients, but there was not really like any logical reason to think that it was going to be in the blood because it was accumulating there or it's expressed in the cells. But we kind of just made a leap of faith there and tried to see if we can identify. And indeed, we found that for some reason, this fragment of NOTCH3 is in the blood, it's in the serum, it's found in humans, it's found in the mice, and the levels change. Actually, we found a decreased levels of the NOTCH3 extracellular domain in the presence of the CADASIL mutation. And what is intriguing is that this is in the blood. Circulating blood has a fragment of NOTCH3 that happens to be the fragment that is mutated in the disease. Now that we felt that we had some biomarkers and that we also can detect some changes in the vessels, we moved to our animal studies. We had generated some animal models for CADASIL. I actually, during my PhD, generated a mouse model that carried the same mutation that we found in this Colombian individual. So I was able to see the patient, and then I made a mouse that carried the same mutation, and now I had the opportunity here of treating that animal model with the drug. So from a personal development and career development, this project meant a lot to me. We had generated many animal models for that, and we also work with mice that are normal for the NOTCH3, mice that don't even have the NOTCH3 at all, mice that express the human NOTCH3 in the absence of the mouse NOTCH3, and we call this a humanized mouse model. Humanized mouse models are great because they are more similar to humans, at least in that one gene. Not everything else, but at least in that one gene, they are more similar to humans, so we call them humanized. And then a mouse that has the CADASIL mutation. In doing this analysis, we focus on the analysis of the vessels, and for that, looking at the retina is quite useful because it has a very stereotyped pattern of the vessels, and the retina is a part of the brain, has a blood-brain barrier, so there are some advantages of doing the analysis in the retina. And this is the distribution of the smooth muscle cells and the pericytes, which also provide support to the vessels in a normal retina. In the NOCA, as you can see, there is a loss of the smooth muscle cells and the pericytes. And I think people have in the field overlooked this, how similar the NOTCH3 NOCA looks to the CADASIL phenotype. Even if you take all the theories aside, just look at that vessel and see the similarities. As I showed you earlier, in CADASIL, what you lose are the smooth muscle cells and the pericytes. The NOTCH3 NOCA that don't even have NOTCH3 has the same phenotype. And what does that tell you? Now, we have the ability using genetics of bringing back NOTCH3 receptor, in this case, the human receptor, into the vessels. And we did this here with conditional transgenesis, and we can rescue the loss of the smooth muscle cells and the pericytes in these vessels by driving NOTCH3 expression. In this case, the human. So the human NOTCH3 can rescue the phenotype of the lack of NOTCH3. But then when we attempt to do that with the NOTCH3 receptor, the human NOTCH3 receptor that carries the CADASIL mutation, there is no rescue. And that is genetic proof that this mutation is loss of function because it cannot rescue the phenotype of the wild. The phenotype, in fact, was identical to that of the NOCA. It wasn't worse, it wasn't better, it was the same. This is a measure of the coverage of the vessels by smooth muscle actin, a marker of the smooth muscle cells, and SMA positive pericytes. So if you express the wild type, you rescue. If you in the NOCA, well, you don't have any rescue, but if you express the mutant, you don't rescue. But it's identical to the NOCA. It wasn't worse. So this is something also to think about. Why is it not worse? if you have this protein-protein interaction or you have this mechanism of toxic accumulation of NOTCH3. One option is that that mechanism is not very relevant for this phenotype. Another option is that we didn't wait it long enough. This phenotype was done in mice when they were 6 months old. So here we have a model that mimics a key characteristic of CADASIL, essentially the loss of the small muscle cells and the parent cells. Another key characteristic of CADASIL is the presence of microhemorrhages. So we're able to show here, this is the retina vessel. And in this method called fluorescent angiography, we can look at leakage events. We identify increased leakage events in the NOTCH3 knockout that we were able to rescue with expressing the human NOTCH3, but not with the CADASIL mutant, but yes, when we had the 2 transgene being expressed. So we had, again, another relevant phenotype to CADASIL, like this is a surrogate for what we call a microhemorrhage. And we're able to show here, these are some electron microscopy images that essentially corroborate our analysis with the immunofluorescence. Now, that kind of just closes the loop. We have a morphological phenotype and we have our blood biomarkers. So we move forward to really testing our modality of treatment. In this case, our hypothesis is that an antibody that can activate NOTCH3 will have a beneficial effect on a CADASIL mouse model. We use our A13 antibody, and here what we did was some preliminary study in cells that express NOTCH3. This is not in the mouse, this is essentially in a Petri dish. But we can show these bars indicate the activity of NOTCH, and when we incubate the cells with the antibody, the A13, we see more activity of NOTCH3, and that happens with the wild type or normal NOTCH3, but also in the presence of the mutation. And here, I don't want to go into the detail, but we're able to show that the antibody has the same effect when we have cells in monoculture just by themselves expressing the receptor or in cells expressing the like. And something here that was actually quite exciting was the fact that we reasoned, well, if the NOTCH3 ectodomain or the extracellular domain can be detected in the blood, maybe we can also detect in the cell culture supernatant, and the levels should change. And our hypothesis was that the levels should increase when we administer the antibody to the culture cells, and indeed that was the case. In the presence of the antibody, we can release, as expected, the extracellular domain of NOTCH3 in normal conditions and also in the presence of the mutation. Now, we felt that now we were ready to do the animal study. We had tested feasibility and efficacy in vitro. We had biomarkers. So we moved to our animal model. In our experimental design, we decided to do systemic administration of the antibody. When people use antibodies, they always have the concern about penetration into the central nervous system. But we knew that in these mice, the blood-brain barrier is leaky. I show you those hemorrhages. Well, that's a problem, of course, to have a hemorrhage, but it could also be an advantage, because if the barrier that impedes passage of drugs to the brain is broken, maybe we can get the antibody out there. So we injected mice at 1 week of birth. We did that every week, and then the animals were analyzed for blood biomarkers and also for the vessel structure. Here is what I was telling you. We label the antibody, and we can show that it penetrates across the blood-brain barrier and gets to where it's needed in the brain. That didn't happen in a normal mouse. What we call the blood-brain barrier blocks the entrance of antibodies and large molecules. And the results were quite encouraging. What I'm showing you here is the retina of a mouse that has the CADASIL mutation but was injected with a control antibody. We call this IgG. And as you can see, the vessel is there. These are the small muscle cells, but they are really covering only the large artery region. There is a lot of vessel there that is just not covered. This shows the whole vessel, but look how little of it is covered by small muscle cells. However, in the animals that were injected with the A13 antibody, what we found it was a completely different phenotype. Here is the coverage with the small muscle marker, and you can tell how there is just a lot more coverage of the small vessels in the treated animals, actually about double the coverage in the small vessels. And this is the quantification indeed in the, we injected the mice that don't have NOTCH3 and we saw no response, but in the mice that had the CADASIL mutation, we found a very significant response, essentially a doubling in the coverage of the small muscle cells. So our interpretation was that this antibody was able to prevent the loss of the small muscle cells, which, as I mentioned earlier, is a key characteristic of the disease. Now, how about the biomarkers? We tested 4 of the biomarkers that we had developed, and 2 of them actually responded. So again, we had shown that in the mice, the levels of the NOTCH3 extracellular domain were diminished in the presence of the mutation, but when we put the antibody, the levels go up, and the result is very robust. With the endostatin, the changes were depending on the age as well. So, and we know that endostatin expression depends upon NOTCH3 signaling. So what we actually observed was an increase in endostatin levels with the antibody injections, which we felt it was a positive response. So with that, we concluded the study for now. Our conclusions is that here we established an unambiguous link between NOTCH3 signaling regulation and mural cell loss in vitro and in vivo. We also showed that we identified these candidate blood biomarkers of cerebral small vessel disease and CADASIL. We developed these in vitro or in petri dish assays to assess the activity of NOTCH3 signaling, and we established the feasibility for a novel modality of treatment for cerebral small vessel disease, specifically for CADASIL. And here I want to emphasize that again, the contribution of cureCADASIL was seminal for the discovery of the biomarkers. The contribution and their support was also essential for the testing of this modality of treatment in mice. Indeed, they provided the funded to start these experiments. And also something that is more anecdotal, but I wanted to share with the community, is that all the work you do, looking online, looking for papers, interacting with scientists could make a difference. The previous president of cureCADASIL, Anne McGuinness, was the one that at one of the first conferences that we have with the CADASIL community brought to my attention the existence of A13 antibody. I didn't know it existed, but she brought it to my attention during one of these conferences. I was just kind of in a rhetorical manner saying that I felt that we had identified a good mechanism and that somewhere someone may have a drug that was helpful for CADASIL, and she indeed had information about that drug and she brought it to my attention right away. So I wanted to end acknowledging the people that did the work in my laboratory, especially Dr. Arturo Machuca. He did a lot of the in vivo work with Alexander Bigger-Allen. These are everybody in my lab that was involved. My collaborators, Dr. Patricia Damore, Margalit Sangenez, collaborators in Colombia and at Genentech, and these are the funding agencies. In particular, I want to acknowledge cureCADASIL for their support, and also something that we started to do and has been quite effective is that people via the Facebook groups, the CADASILians Facebook group and the CADASIL Support Facebook group have also supported my lab with their donations. And with that, I end. So I pass it on now to Jane, that is gonna lead the Q&A session.
All right. Thank you, Dr. Joe. It was really educational. And thank you for all the work you have done with CADASIL over the last 20 years. We'll now give you, there's 4 questions, and I'll give you the opportunity to answer each one. The first question is, what are your thoughts on research published a few years ago suggesting leaky vascular basement membranes as a cause of CADASIL?
Can you repeat the question again? Sorry.
Yeah. What are your thoughts on research published a few years ago suggesting leaky vascular basement membranes as a cause for CADASIL?
Oh, good. So that's a really good, interesting question. I put the webcam if people are interested. Maybe you know, I thought that we can like make it more interactive, and I have a board in case I need it. So the question refers to the basement membrane. We talk about 2 components of a vessel. One of them was the endothelial cell, which forms the lumen. You know, inside you have the lumen of the vessel, and then you have here the smooth muscle cells that we focus on, and the pericytes. Well, in between, what you have is what we call the basement membrane. The basement membrane is the matrix that connects and holds the cells together. So indeed, it has been shown that the basement membrane plays a very important role in small vessel diseases in general and in CADASIL. There are differences in the basement membrane composition between normal vessels and CADASIL vessels. There is not just a process of cell loss, but also a degenerative process in the basement membrane. I think that basement membrane and matrix alterations are key to the pathology. They contribute to leakage, and they contribute to vascular degeneration. Now, it's difficult to target the basement membrane as a in a therapy, because the proteins that are made there are deposited. What are you going to do? Are you going to try to have cells make more matrix or less matrix? So it's I think it definitely plays a role, but it is also important to recognize that, in my opinion, it's difficult to target the basement membrane and as a therapeutic. People have tried, and there are efforts toward that.
Oh, thank you. It was very nice to have that drawing. Our next question is, do non...
Let's see how it goes. Go ahead.
Do non-EGFR domain Notch3 variants cause CADASIL?
Oh, okay, very good question. So that question is, as I mentioned earlier, this is a an schematic of Notch3 receptor in the plasma membrane. All of the known and accepted CADASIL mutations happen here in the what we call the EGF-like repeats. Okay, so the mutations that are accepted happen there, and this is what we call CADASIL. Now, this term might not, or this terminology might not be sufficient for what we actually observe in humans. A CADASIL diagnosis, as it was classically defined and is accepted by the doctors and people that are interested in terminology, is that the diagnosis requires that the mutation is in Notch3 and that the mutation affects the system and that the mutation is in the EGF-like repeats. Now, people are finding other types of mutations that impact NOTCH3. And those mutations don't affect system, so the system is not affected, or they are outside the EGF repeats. So the EGF-like repeats are okay, but it's in a different region. Or there are mutations that actually make the gene not to be expressed. They are a true loss of function mutation. So the mutation will be like a loss of function. In that case, is what we call like a stop codon or a frame shift. The gene is not made there. Now, in some of these individuals, they have a disease that affects the brain, is often inherited, obviously, in an autosomal dominant pattern, is characterized by arteriopathy and leukoencephalopathy. The patients get infarcts in their brain. Really, they check every box in terms of the pathology, but they don't meet the requirement for CADASIL diagnosis. And this is why I'm proposing in the field a shift from a deterministic view of the CADASIL diagnosis to something that is more useful to patients and also more useful for people to understand the biology. I will call that NOTCH3 associated cerebral small vessel disease. So in that case, if we are talking about NOTCH3 as the gene that is important for vascular stability, if we have mutations that affect NOTCH3 and they are demonstrated to be pathogenic, those mutations can cause cerebral small vessel disease. And that is a more generic term that will include CADASIL and also will be inclusive of what we're seeing in other patients that have similar phenotypes, but have different types of mutations. So I think that would be, and I think the field is moving hopefully in that direction, because we have a lot more people that have mutations in NOTCH3 and they have similar symptoms and pathology. So I don't think that that view is necessary, is a moving target, but I think we will end up there. Is something that I will think will be a more comprehensive view of the disease that we see in the patients.
Very interesting. Thank you. Third question is, have you found a connection to immune system dysregulation and CADASIL?
What was that? Can you
has there been found a connection to immune system dysregulation and CADASIL?
Okay, so in that case, I will say that there is more research that needs to be done on whether the immune system is dysregulated in CADASIL or not. Some people will say, well, do you have inflammation in the vessels of CADASIL patients? The answer to that is no, at least not very obvious. But does the immune system play a role? Well, why do we have so much variability in the clinical presentation of the disease? Could it be that the immune system and the differences in the immune system between different individuals play a role? If the disease, for instance, is working via a mechanism of protein accumulation, well, the immune system is excellent at clearing proteins when they accumulate. That has been shown in Alzheimer's disease, and that has been shown to explain some of the phenotypes in Alzheimer's disease. So could it be that in CADASIL, the different presentation of the disease, why some people get stroke at this age or at that age, why people get more dementia than others, could that be explained by changes in immune system and the ability, for instance, of develop naturally occurring antibodies? In this case, we are not talking about a drug that we inject, but naturally occurring antibodies against NOTCH3. So I will answer that question by saying that more research needs to be done.
Okay, thank you. Are there any, this is our last question, are there any studies being done or considered as far as the effects of stem cell treatment for CADASIL?
Stem cell treatment? Okay, so
Yes.
that's very, you know, I think that's a very important lead for the following reason. If in many CADASIL patients, if some of them could be listening, if you're in your 40s or in your 50s, some cells have been lost already. So how do we get those back? And I think we might be in a, and then one way to do that is with the stem cells. Hopefully you can have a situation in which you can prevent some more cells from dying, but then via stem cells, you can actually repair vessels using stem cells. And you can do that by promoting a stem cell regeneration using factors, and there was a talk about that. Or you can do that perhaps by, you know, injecting cells. I think that there has been some progress on that, not specifically for CADASIL, but there are people trying to do that, like applying stem cells to like regenerate the pericytes or the small muscle cells in a vessel. And people have shown that it could work in animal models. It hasn't been, to my knowledge, tested in humans, but theoretically it could be done. So I think it's a very important area of research. And I got a question here also. They are asking me if we have time to answer it. I will say yes. I don't know if you also see it, Jane, and you can read the question, or if I should read it.
Go ahead, you can read it.
Okay, so the question is, there really wasn't rescue with the wild type. At best, it was a very partial rescue. Why? That is a very good point. And I think what we show when we did the genetic rescue was that there was a significant increase in the vascular small muscle cell coverage in both the large vessels and the small vessels, and that's what we call a rescue. It was a significant rescue. Now, it was not a complete rescue, and we believe that that is related with the expression pattern of our driver. People, when we think about conditional transgenesis, you have a gene that could be expressed and another one that drives, in this case, clear recombinase. Now, the effectiveness of your rescue will depend on two things: whether your gene is effective at doing the rescue, and whether you are driving your recombinase in an enough number of cells. So at this point, I think that the partial rescue could be related to that. For instance, we got more rescue, I think, by in the large vessels compared to the small ones, and I think that has to do with the fact that the driver that we use is more expressed in larger vessels compared to the small muscle cells than pericytes. But then, and that finding is consistent with then what we saw with the antibody. The antibody doesn't have that problem necessarily, and we found more rescue in the small vessels compared to the large ones. I think more research needs to be done. It's not just, well, it was partial and that's the result. There are other drivers available, like Cree drivers, to do the recombination. For instance, the small muscle myosin can be used for a pericyte specific. I think what we showed was proof of principle. It could be rescued, but definitely more research needs to be done to see the extent of what is possible.
All right.
Anything else?
Thank you for taking that additional question, and thank you everyone for your questions. Very interesting questions, and really brought up a lot more information and things for us to think about. So thanks again, Dr. Joe.
Bye-bye.
In closing, our goal is to connect the CADASIL community. Please contact info@curecadasil.org if you would like more information from our speakers or if you have a topic of interest. One suggestion we have received is to announce our volunteer opportunities, and posted here is our short list of immediate needs. Please contact us. We look forward to meeting and working with more people in the CADASIL community. We hope you can join us at our next webinar with Dr. Michael Wang. The date will be in the fourth week of April, so keep an eye out for when that date is set. The recording of today's webinar will be available at our website. Thank you to Dr. Joe for the time he has donated to advance biomarkers and the therapeutic in CADASIL and to make today's webinar a success and very interesting. Thank you for your participation. This is the conclusion of our webinar.